question. 8. So you mentioned how plants had a less defined body plan compared to like animals. Can you explain why again?
Can you repeat your question, please?
You said that plants had a less defined body plan compared to like animals. Can you explain why?
Yes, give me a sec. So as we can see here, what happens here is the animal cell goes into the ectoderm, mesoderm, and endoderm, and from there we have like more cell specialization. In terms of the body plan for animals and plants, it's a good question.
Yeah,
Thomas, do you have the answer for this?
I'm not too sure how to explain that either. Because I mean, like, oh.
So why do animals have a more specified body plan than plants?
Yeah. That's kind of the pluripotent part. After a number of subdivisions and then basic maturity, the cells are just into the plate to make sure that organ is fully developed.
So just like adding on to that, I'm sure you guys know, like just by reading the lectures, there's a lot that we don't know about cell biology. So even the interaction part, even the cell specialization slash differentiation part that I discussed, this is a theory, right? So it hasn't been like it hasn't been like established. So usually like going off of what Albert said, cells usually specialize to their function. So epithelial cells tend to endure more stress, right? And then they tend to be like more stretchy. So form fits function basically. And there are some early signals during cell division, so cell proliferation, cell specialization. There are some early signals that signify like where these cells should go. To be honest, I don't know like what exactly is the mechanism for cells like knowing exactly like, hey, I'm gonna form like a nerve cell versus hey, I'm gonna form a skin cell and I'm gonna go right here. But Albert's answer of it all comes down to genetics is absolutely right. Like it hits it on the head perfectly because um, there are going out of time. Okay, but yeah, genetics. That's the answer.
Thank you. Thank you.
Yeah, I think so. Yeah, I think so.
Yeah, I'm ready. All right, perfect. So our presentation is on the Hawk system, which is also known as the Hawk genes, which is a perfect segue from the previous presentation, which had the body plans of differentiations of body plans between plants and animals. So on the first slide, we have what are Hox genes? So Hox genes are also known as homeobox genes. They are a subset of homeotic genes, and they are a group of specialized genes that maintain a general blueprint of the animal's morphology so that the correct parts grow in the correct segment. These genes are the most utilized during homeotic development, and if you look at the image on the slide, there's a mouse embryo and an adult human. The HOX-A genes are in charge of the thoracic area, while as the HOX-D genes are more of the um, like lower body type legs, arms, limbs area, and AB are part of the cervical, like the cerebral type area. And 39 human Hox genes are located in 4 different clusters, like the ones I named before, A through D on different chromosomes, and mouses and humans have similar Hox A through D genes. The importance of Hox genes are because they regulate homeotic development of the anatomical structures in a number of different types of animals, like insects, mammals, plants, and even ectoderms. They aid in uncovering evolutionary discoveries like analogous structures and convergent evolution between animals. So if you view the picture, the image on the bottom, there's a shark fin between the penguin fin and the dolphin fin that helps with analogous structures are similar, but they follow the same similar body plan. And when morphological mutations occur, it is due to an incorrect transcription of how the genes of the genes, which can lead to anatomical malfunctions. So the above picture is a picture of a fly, and instead of having the picture on the right has the legs instead of the antennas, which means that it's makes it more difficult for the fly to navigate and move around on surfaces.
Can you hear me?
Yeah.
Okay, cool. Just making sure. Okay, so what are the differences between insect and vertebrate Hox genes? So basically, there are a few key differences between them, the both. So the first one is the number of clusters. So for instance, insects have 1 and vertebrates have 4. So as mentioned earlier, if you look at the diagram, home C is the insect's one and only clustered chromosome, while vertebrates have 4 clustered chromosomes: Hox A, Hox B, Hox C, and Hox D. The number of individual Hox genes. So insects have 8, vertebrates have 39. So basically, what that means is on each one of these individual clustered chromosomes. So for HOMC, for insects, there's 8. So basically, each one of those individual square color boxes is a Hox gene. So there's 8 of them total for insects, and for Hox A, B, C, and D, that all adds up to a total of 39. The number of genes per chromosomes. So since insects only have 1 clustered chromosome, they only have 8. But for vertebrates such as humans, so for Hox C and D, each one of those Hox genes, they add up for those two 9, while for Hox A can add up to 11. And then you can go. Yeah. I think because it has to do with the because for vertebrates, I was kind of get to that a little bit in the next slide, but basically it has to just do with more shaping and the structural form. Yeah, okay, so for insects, their Hox genes specify for appendages to form particular segments. So basically, the legs need to go where the legs need to go, and so forth for the rest of their appendages. So for vertebrates, their Hox genes specify the type and shape of vertebrates that will be formed. So I feel like that's probably specifically why there's way more Hox genes, because it's kind of more intricate and not so much putting appendages where they need to go, but more for the type and shape of the vertebrae. And it's believed that duplication of a single clustered chromosome, they duplicated it into 4 other evolutionary in history of vertebrates. So basically, Hox genes, the Hox A, they're speculated that it duplicated itself into 4 clustered chromosomes. So into Hox B, Hox C, and Hox T.
Hox genes direct the body plan along the anterior and posterior axis of the organisms. They contribute to the expression of different animal genes shown in physical characteristics. For example, the number and position of limbs on an animal or the color and texture of their skin. This picture here is a demonstration of the segmental identity. It shows that Hox genes can determine whether a fly will have 2 or 4 wings and where they're located on the animal's body. Hox genes encode homeodomain containing transcription factors. These Hox genes are turned on by a cascade of regulatory genes. In this cascade, the proteins that were encoded by early genes regulate the expression of later genes. Altering Hox proteins. For example, in fruit flies, if you alter the Hox gene ultrabithorax, it results in change in the segmental identity seen as another pair of wings, like in the previous slide. This is usually done during the developmental stage. Hox genes turn on in specific patterns because of the protein product of gap and pair rule genes, which are genes that divide embryos into pairs of segments. The interactions of Hox proteins is when the original identity of the segmentation of the organism is changed.
So, is there signaling involved in Hox genes? And the answer is yes. There is multiple types of signaling involved in Hox genes. Hox genes are first activated within stem cells and are then encoded to transcription proteins. The image is the activation of stem cells when signaled by Hox genes. Predominantly, the posterior 5' gene of the Hox gene chromosome are activated by the signal factors BPM and WNT mainly. The signals go through separate signals in order to accurately transcribe and activate Hox genes. So in short, Hox genes do involve signaling, yes, but it's done little by little because it's crucial that the blueprint of the body's anatomical structure is followed. But as always, there are some mutations and some mess ups in the transcription. And since Hox genes are responsible for the entire blueprint of an animal's morphology, it has to be able to signal to different sections of the body. So in the image besides it, the different signal sections include axial elongation, which elongates the length of the body, P identity, which specifies the anterior posterior axis, and mesoderm segmentation, which segments the 3 primary dermal layers, and if applicable, since ectoderms don't have mesoderms. Yeah, so is there any questions?
I had a question, but I think it was kind of similar to yours, Albert. And uh, I'm not sure if they answered it or not, but it was what was the significance between the different numbers of Hox genes in insects and vertebrates?
Yeah, go to the next one. So so what I think it is so basically for insects, there's so few because all those genes really do is specify what appendages go where. So basically tells the legs should go to the legs and the wings should go to the wings. And then so for humans, it's more intricate in it specifies the type and shape of the vertebrae that needs to be formed. So I feel like it's just more just I don't know, just more intricate and more to it. I guess. I mean, Albert, you want to help me out here? I guess maybe because I mean, because it was very vague. I mean, I was looking, and it because most of it does practically similar. They're very similar to each other. I think there's just more in depth and there's more to do because it's a bigger, I guess, unit maybe.
I have a question. Can you hear me?
Yeah.
Okay. I was wondering if you could maybe clarify again the I think 7. But I was wondering if you can give some more background as to how the Hox genes evolved into the clusters, like in the picture. You were going over like the history of vertebrates and everything, but I didn't quite catch that.
Wait, so what was your question exactly?
The line that says duplication of a single cluster, I was wondering if you could maybe provide a bit more background
Yeah,
on
so
that.
yeah, so basically what I was saying is um so for vertebrates it kind of was like like evolutionary before in history it kind of it did start off as just one cluster, and then what I was just saying is throughout history it replicated, duplicated itself into onto four other chromosome clusters. Is just basically what I was saying is it started as one, but it duplicated itself into four for I guess because it was just a more complex structure and it was more work for it.
Yeah, and that really correlates with what Albert said about the why humans have more or animals have more chromosomes for the Hox genes. It's because of the duplications and also it's a more complex structure and unit. And if you think about it, comparing an animal specimen to a fly, it's a lot more intricate, a lot more things going on, and certain body parts and body plans need to go accordingly. And if you think of, like, let's say a fly has two wings instead of four, and a human has one arm or like missing limbs, like it needs more instructions in order to um go according to plan.
Thank you.
Mhm.
Albert, can we just get like a minute or two to like go through the evaluation sheets, just so we don't forget what we saw and heard and stuff? before we move on to the next group? Thank you.
I had a question. My question is, you mentioned that there was signaling in the gene. Did you mention what types of signaling?
It was really broad. Most of it was translation based. So it was like the basic central dogma type signaling where it went from it grabbed it created proteins and sent gene information from cell to cell. It was really vague, like this diagram. So sorry, I need to open it on a different page because it's too small on this one. It's like, so this diagram basically it's all the transcription and activation signals being sent through the cell wall and sent to the chromosome, and it starts from the 5' chromosome, and then slowly it goes back and forth through the nuclear envelope to send the proteins out and in, because like I said before, it's a really complex chromosome that we need, that the body stresses to get right. But like I said, there's always mutations and whatnot. So it works its way down and out of the nuclear envelope to make its way from the 5' Hox gene to the 3' Hox gene. Yes. Yes, correct.
Spencer.
Need like 2 minutes.
Okay, we're ready. So our group did a presentation is on the Arabidopsis, Sam, which is the Arabidopsis shoot apical meristem. So a little bit of background on the meristem. Next slide. The meristem we're focusing on is the Arabidopsis thaliana, which is a plant species that's specific to Eurasia and Africa. This specific species is used as a model in molecular biology due to the fact that it has approximately 140 million nucleotide pairs and that it gives off so many offspring and it reproduces fast. The shoot apical meristem is typically like the top part of the plant that shoots out. It gives all the organs like leaves, stems, and flowers. The shoot apical meristem typically determines the fate of organs. Early on, so it's early determination, and it can also regulate and maintain the growth of these organs through different genes and receptors. So an example of how it would maintain it, let's say that the plant is in an environment where maybe it's too dry or if it's covered by certain rocks, the shoot apical meristem will detect that and either suppress growth or it'll find a way to go around it. So it could be like if there's a rock, it'll find a way to grow through the cracks of the rocks and shoot up through into the top of the roots.
Oh, hello. Can you guys hear me? Sorry.
Yeah.
Okay, so typically plants exhibit 3 layers of SAM, also referred to as colonal layers, and they're distinguished by the planes of cell division. So tunica makes up the first outermost layers, and these cells divide anticlinally, or dividing in an upward arch, so as shown by this diagram on the right. And then the next layer is called corpus, which is the next layer underneath the 2 first ones. These cells differ in how they divide. They first divide preluminally and then divide both ways, anticlinally and periclinally. Sorry, my pronunciation is pretty bad. But basically, this layer serves to add bulk to the shoot, widening the plant's apex so they grow side to side. And I guess all these diagrams further display these processes. Oh, it's not going.
Okay, so like all living things, plants are subjected to mutations. In this particular plants, mutations are due to the mutations in the West gene.
Oh, hello. Oh, they show basically how these cells divide. So if you look at this one, that's B and it has the green diagram, it shows how a lot of these cells grow side to side. So first they would grow up and then they would grow side to side as widening the plant's apex. Yeah, well, that's how you say it. Oh, so the inner layers, they divide both anticlinally, yeah, and then they also divide periclinally. Yeah, sorry, I'm like really bad with my pronunciations, and I probably should have listed it on the slide. But they, like I said, they add a variety, so they both grow up and out. So basically, anticlinally, I mean, it grows in an upward arch. And then the other one would be growing side to side and adding bulk to the plant. Yeah. Yeah. Yeah.
Okay, as said before, mutations are due to the mutations in the WES and CLV genes. In the WES, this has to do with regulation and maintenance of the shoot apical meristem, and the CLV controls plant populations. So as you can see, as you can see in the bottom of the screen, you have the wild type shoot apical meristem, which is what it's supposed to look like. One mutation would be the shoot meristemless. So this happens when the shoot apical meristem is reduced or completely absent in mature embryos. The next mutation would be the pinhead one. This particular mutation is a result of when the lateral organs are curling upward with the leaf blades. This has to do with the loss of cell number coordination between the two surfaces of the blade. And then the last mutation we have here is a cup-shaped cotyledons, which is a defect in the separation of the embryonic organs, cotyledons. Also in the floor organs, the sepals and the stamens, which will mess up the organization of the shoot apical meristem.
So like it said on the previous slide, most of these mutations are caused by the Wishel genes and the clavata genes. Specifically, the Wishel genes, that's a homeobox gene that is key in plant cell organization and maintenance, basically being responsible for cell state. So a mutation in this gene can lead to plants being unable to self-organize and unable to make effective meristems, eventually leading to them terminating prematurely in flat structures. And this is displayed by the left picture right here. I guess these mutants would have no true bud in the middle of the plant where the flower would be. And then the second set of genes would be clavata genes, and these are responsible for controlling the cell populations within a plant's SAM. And there are three main genes here: clavata 1, clavata 2, and clavata 3. The first two function as receptors, and the last one functions as a ligand. So a mutation in any of these genes would result in an abundance of meristem cells at the apex of a plant, and the plant's shoot unable to grow any further. And this is displayed by the diagram on the right. This would be the wild type with, I guess, the right number of petals and a controlled cell growth. But E and F obviously show too many petals and just, I guess, a different type of plant. Oh, yeah. I guess it was explained in one of the previous presentations, but a homeobox gene that would be key in, I guess, providing a type of structure or a type of structure for how the plants, I guess, grow. Yeah. Oh, okay. Okay. Well, clavata and West are considered, or clavata and Wischel are considered to be couple reactions. They're both controlled by feedback signaling, which in turn maintain a plant shoot apical meristems. In this process, regulation of clavata leads to the inhibition or of Wischel, allowing for cell differentiation to occur. To be more specific, the clavata 3 genes, like I said before, serve as a ligand, and clavata 1 serves as a receptor. Their binding leads to phosphorylation of proteins, thereby activating a row GTPase. Wischel normally inhibits the transcription of genes required for differentiation. So by blocking it, cell differentiation occurs, and so is the ability for continued growth of the shoot meristem. The first diagram here, the top picture shows a normal wild type with a typical, or with the bud plant. Oh shoot, with the bud in the middle. And the picture below shows what would happen if the Wish-Chill gene were to be activated. There would be no bud in the middle. And the middle diagram shows just how this is supposed to, how these genes are supposed to be interacting. Activation with clavata means which means the blocking of the Wish-Chill gene. Do you have any questions?
I think on the next slide.
Yeah.
So for the mutations in the clavata, it produces extra like floral organs and enlargement of both the shoot and floral meristems. And then, so in other words, it makes them bigger. And then mutations in the Wuschel development, it would make the seedlings fail to sprout or make them flat in structure. Was that the question, or was it different?
Well, from what I've read, I guess the book wasn't too helpful, so I had to do a lot of research online. But basically, clavata is a set of genes that are responsible for controlling or for basically giving the plant or the cells within the plant a structure to follow. And then I'm sorry. And then the Wish-Chill genes are basically, like I said before, the key in plant organization and maintenance. And when they are activated, it results in determination of a meristem within a plant. So like in the pictures I showed before, let me see if I can go back to the other slide. There is no bud growth here, but yeah, if it's activated. But if it's not activated and kept blocked, the gene, then the bud would still grow, like in the wild type. No, clavata leads to, or Wish-Chill activation of this gene leads to the stem or the meristem termination. So it's activated in let me go back. It's activated in, I guess, the corpus layer. Right? Yes. Right here. The bud in all the pictures that I found online and everything that I read, it showed that basically there was no bud growth here. It led to plant termination. All this little cells right here would be terminated within the bud, so it wouldn't be able to grow. Yes. Yeah. Oh, okay. After the presentation, can you maybe send the slides? Because like I said, like we've been working on this presentation for a while and we couldn't find really any information on it.
So some supporting genes of the shoot apical meristem include the MIR156. This will aid in the regulation of the shoot apical meristem in terms of early development and growth. And in terms of Arabidopsis, thaliana, it also needs the MIR159 gene, which will help to determine the exact timing of vegetation growth. The SPL gene, the squamosa promoting binding light gene, aids in meristem size and the juvenile to adult vegetation transition. The LRR gene, the leucine rich repeat receptor kinase gene. There are about 175 different LR genes encoded in the Arabidopsis genome, but it regulates growth, development, signal transduction, immunity, and stress responses in the plants throughout its whole life cycle. An example of that would be the BRI1 gene. This particularly regulates the growth and differentiation of plants. It's a cell surface steroid hormone that is commonly known, or most people will know it as a brassinoid steroid, and it's actually in the same family as a Arabidopsis. So without this, the plants wouldn't grow and it would end up being a dwarf.
Both the westral and the clavata are expressed in different layers of the cell. So in L1 and L2 layers and part of the L3, it contains cells for the meristem proper. And then in L1 and L2 layers, the clavata 3 is expressed, and it's a signal protein. So just under the layer 3 is the cluster of cells expressing clavata 1 in the center. So in the center is a clavata 1 patch where the cells express the westral gene regulating protein. And the westral cells release a signal that stimulates the expression of the clavata genes. And this causes new cells to be recruited into the westral domain to switch on westral. And then so there's a negative feedback from the upper meristematic cells derived by the clavata signaling pathway. And so this makes sure that the meristem doesn't become too big. Then the next slide. Can you switch this slide? Sorry. So this shows us an important developmental strategy. It shows us a feedback loop that involves both the short range activating signal and a long range inhibitory signal can maintain a signaling center. And although this shows it in plants, we can learn a lot about it, the similarities it has with animal cell development as well. But a key difference between the two is that in plants, the transcriptional factors are able to travel, while in animals, they can't.
I'm sorry, could you repeat that? This one?
Yeah. So, I think it was. So I think it's when once it releases the signal, Once it releases a signal, I think it turns off the west shell. Yeah. Yeah. Mhm. Yeah. Yeah. Oh, yeah.
I have a question. Groups. Let me double check. I'm sorry. I think I'm group 6. Yeah, group 6. Um, so does the Arabidopsis SAM genes only apply to Arabidopsis family, or are they applicable to other plant families as well?
Can you repeat that question?
So does the genes only apply to the Arabidopsis family, or are they applicable to other plant families as well?
They are applicable to other plant families, but we just singled out the ones that were in the Arabidopsis family.
Okay, perfect. Thank you.
I have a question. I'm group 1. So what happens if SAM is shut off during development? Will the plant like not grow properly if there's a rock on it, or not have directional cues?
So, as we said earlier, in terms of the mutations, if SAM wasn't developed properly, it could lead to dwarfism. It could lead to certain organs not being developed at all, or maybe developed improperly. And in terms of the rock example I gave, if it like senses like light, for example, it'll grow out to where it finds light stuff where it finds a surface. Not all the time. It's in different cases. Not all the time will a rock stop a plant from growing. It'll start going through the cracks. If you can see like during like or through rivers and stuff, you start seeing like plants go up through the rocks and through branches and stuff. It's like that. Does that make sense?
Yep. Thank you.
I have a question. Group 4. I think that one of the biggest factors for plants is their environment. So, depending on like when you guys talked about these mutations, like these pictures are showing like to only affect like plants that grow in like a moist environment compared to like desert plants. So, like, do these mutations also impact like those desert plants also?
I believe it would. It would probably impact it maybe a different way because desert plants sometimes grow differently than like a plant in the water. But yeah, with for example, with the mutation in the cholesterol gene, it has to do with the regulation and maintenance of the shoot apical meristem. So if there's a problem with it and it causes a mutation, regardless, organs are going to be messed up or they're not going to grow.
All right, thank you.
Mm-hmm.
I had a quick question. Can you go over the different layers of the shoot apical meristem again? Group 2.
You mean this slide? Hello?
Yeah.
Yeah, so basically the two outermost layers, I guess it should be, yeah, that would be called the tunica. And the one layer underneath would be the corpus. Did you want me to go into anything specific or?
That was it. Thank you.
Oh, I'm sorry. Yeah, it's so L1 and L2 within this diagram are supposed to be the tunica layers, which makes the two outermost layers. And then those are the ones that divide, like I said, an upward arch. And then corpus would make the third layer underneath, all that's in blue and all that's in dark green. And yeah, those are the ones that provide the bulk of the plant, like I said before, dividing both an upward arch and I guess perpendicular, yeah, side to side. I'm sorry, could you repeat that? I personally didn't read too much on that, but I'm guessing it's because the first layer, L1 would be the outermost one exposed to all the elements, and then the second one would just be the one underneath. Yeah. Sorry if that doesn't really answer.
Okay, hi everyone, we are group 9, and today we'll be talking to you about mitosis and cell cycle. I'm Adriana, and I will be discussing the purpose of the cell cycle and mitosis, the stages, and the cell cycle events. Cyrus would discuss the mitosis event and cytokinesis, and Lizette would discuss the sister chromatid separation biology and regulation checkpoints. So let's begin. The purpose of a cell cycle is to regulate the production of cells, as well as for the activation and inactivation of DNA synthesis and replication. Now, this regulation is an important step to lead to the activation of mitosis. The cell cycle involves the replication of chromosomes, cell growth, chromosome segregation, and the cell division. I'm going to give you a little background on mitosis. Mitosis was discovered by a German anatomy biologist named Walter Filming in 1887. The mitosis term comes from the Greek language, which is translated to wrap thread for the mitro part, and oasis ending is the act or process. The purpose of mitosis is important in all eukaryotic cells, such as plants, animals, and fungi. The three main purposes for mitosis are the ones listed below: developmental growth, cell replacement, and asexual reproduction. Developmental growth occurs after meiosis has produced a gamete and forms with another gamete to create an embryo, which then this embryo will use mitosis to grow. The cell replacement occurs when an original cell that has been damaged or wounded will have new cells to be created to replace those that are damaged. An example of this would be healing from a cut. And finally, the asexual reproduction are those that use mitosis to reproduce asexually, and we know these to be single-celled organisms and certain multicellular organisms. The stages of the cell cycle consist of interphase and the beginning of mitosis. Now, interphase has three main phases: G1, S phase, and G2. Mitosis has its own course of phase that later Cyrus would go more in depth soon. But these phases are prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis known as M phase. Now for the cell cycle events, as I mentioned before, it takes place in the interphase, and the interphase contains G1, S phase, and G2 that follows. This is the process of where cell grows and prepares the cell for division. The starting phase is G1. This phase is the starting point of the cell cycle. It consists of setting up the cells into a committed step. It does this by ensuring that the cells contain the right nutrients and are the correct size to proceed. But the G1 cell phase is also where the product of cycling proteins are made. Cyclic proteins will be able to activate kinases known as cyclic dependent kinase to carry out and regulate the cell growth. The following phase that follows after G1 is the S phase, which is very important for the starting of the DNA replication. To prepare our cells for cell division, chromosome duplication must occur, from 23 pairs of chromosome to 46 pairs. This process is activated by cycling protein A that will form a complex with cycling-dependent kinase 2 in the S phase. The following phase, the G2 phase, it is known as the transition checkpoint. It is in charge of double-checking the genetic information from the previous phase, such as if the DNA replication is done correctly or if the DNA is damaged at any point in order to proceed to the next step. If there is a possibility that the DNA is damaged at any point, then the damaged DNA will not be able to continue and it will be held to be repaired. However, if there is no damage found on the DNA, then the process will continue and mitosis will be activated. Mitosis is activated by another cycling protein. This one is known as cyclic protein B that will form with a cyclic-dependent kinase, in this case, cyclic-dependent kinase 1. Now, the following phase of all genetic information, is sent to complete the cell division in the M phase. The M phase is the process that chromosomes segregate and cytokinesis occurs. And once again, it is divided into 5 stages: prophase, metaphase, anaphase, telophase, and cytokinesis. The last event of the cell cycle that I will be discussing is the G0 phase. This phase is technically the end of the cell cycle, and it is known as the resting stop for cells after meiosis. In this phase, the cells will no longer be able to divide, and they can either be put to store in this phase for later usage if the organism needs repair, or it will become a mature cell and exit to become a heart cell or a neuron. G0 will be that little black line between M and G1. It's not present on this diagram, but it's right in between.
Alrighty, so I'm going to be going over the stages of mitosis, which we're going to start from interphase and all the way down to telophase and cytokinosis. So the first stage of mitosis is called prophase. That's when the replicated chromosomes that contain two closely associated sister chromatids condense. And then outside of the nucleus, the mitotic spindle assembles between two centrosomes, which have replicated and moved apart. The second part is called prometaphase. This starts very abruptly with the breakdown of the nuclear envelope. The chromosomes are now able to attach themselves to these spindle microtubules via their kinetochores, and they go under active movement. The third phase is called the metaphase, and during this phase, the chromosomes are aligned at the equators of the spindles, midway between the spindle pole. The kinetochore microtubules attach to these sister chromatids, which are on the opposite poles of the spindles. The fourth phase is called anaphase. The sister chromatids separate to form two daughter chromosomes, and each is pulled slowly towards the spindle pole it faces. The kinetochore microtubules get shorter, and the spindle poles also move apart. Both of these processes contribute to the chromosome separation. Lastly, telophase is the final stage of mitosis. It's when the two sets of daughter chromosomes arrive at the poles of the spindle and decondense. A new nuclear envelope reassembles around each set, completing the formation of two nuclei and marking the end of mitosis. And then the division of cytoplasm begins with the assembly of the contractile ring, which I will talk about in cytokinesis. So cytokinesis is completed after telophase, which is the fifth phase of mitosis, which marks the end of cell division. This phase is the result of the membrane of the parent cell that's splitting into two daughter cells. And it's important to the cell cycle because not only does it complete this cell division of mitosis, but it also ensures that each nucleus ends up in each of those daughter cells. And it's also a process that will send signals to start this new cellular process. And cytokinosis usually has 4 different stages: initiation, contraction, membrane insertion, and completion.
Hello. So sister chromatids, they do separate in the cell cycle, and they separate at the metaphase to anaphase transition. They separate by a protease called seprin. Seprin cleaves a protein called cohesin, and basically cohesin is what holds the sister chromatids together. Seprin cleaving cohesin is what allows the chromatids to separate. So the cell cycle does have, or the cell cycle is regulated by checkpoints. These checkpoints are basically in the cell cycle just to make sure that each step is done and it's done completely and right in order to continue to go on to the next stage. Checkpoints are internal decision-making processes that can be found within the cell cycle. So first we have the G1 checkpoint. It's also known as the start restriction point. It's the commitment point and it makes sure that the nutrients, size, and growth factors are okay for the cell to enter the cell cycle. Next, we have the G2 and M transition checkpoint, and basically it makes sure that the DNA is replicated and that it's okay and not damaged to continue on to mitosis. And then we have the MA transition checkpoint, and that basically makes sure that all the chromatids are attached and that cohesin can be cleaved to separate the chromatids and then continue on to cell division and complete cell division. So overall, the cell cycle results in cell division. It involves the duplication and separation of cell constituents. The cell cycle is regulated by many checkpoints, ensuring one point or one event is complete before the next event begins. A cell does decide whether to divide or not, and that depends on environmental controls such as cell size, nutrients, and protein growth factors. And in yeast cells, it's up to the cell size and the nutrients. And for mammal cells or mammalian cells, it's up to the protein growth factors. And that's it.
I have a question for group 3. Hello, Albert. Yeah. Okay. You mentioned a lot of cell cycle checkpoints in the G1, G2 phases. What would happen if a cell that wasn't supposed to pass the checkpoints, like, passes it anyways?
I think those checkpoints are there so that it doesn't continue. So if it wasn't okay to continue on, that checkpoint would make sure it doesn't continue on to the next stage. Yeah. Well, like I read that like when cell division isn't regulated correctly, it can lead to like a lot of cell division, and it can lead to things like cancer, if that's a good example.
I have a question. If the what happens if the regulation checkpoints are broken or missed, so the cell cycle just doesn't continue?
Sorry.
Group 1.
Can you repeat that?
Yeah. So what happens if regulation checkpoints are broken or missed?
I don't think there's a. Oh, go ahead.
So I was gonna just say the way these checkpoints work, it's kind of like a stop start. So after the completion of each phase, you stop at these checkpoints and then you start the next phase. So if you don't complete these checkpoints, you won't be able to start the cell cycle.
Okay. Thank you.
Right.
Yeah.
Yeah.